quantitative big imaging. Full screen. Okay, so the slides and everything should already be online. There were some changes made to them and a few slides from last year that will be put in there, but all the links are on the page, and so they should work fine. So this lecture will be about basic segmentation and discrete binary structures. And so what that connects to, we'll sort of talk about in the beginning. So what's the initial motivation for doing this? What are some of the kind of qualitative approaches? What is thresholding? What other kinds of images can you apply it to? What does sort of selecting a good threshold mean? And then we'll talk a little bit about the machine learning. Has anyone taken a machine learning course already? 2, 3, 4, okay. So a few of you. Okay, so, and does ROC curve mean something? Who's never heard of ROC curve before? Okay, so we'll have to cover that. But if you've heard of it before, it'll make that a little bit easier. And it's a very useful technique in general when you're dealing with sort of classification style problems. We'll then talk very briefly sort of about implementation. The point of this class isn't to get into too much detail about that. And then a bit about morphology. And that's sort of where we'll leave it. So the applications for this are really kind of simple two-phase materials, bones, cells, beyond one channel of depth, sort of multiple phase materials, filling holes in materials, segmenting fossils, and attempting to segment the cortex in brain imaging. So that's kind of the examples we'll go through over the course of this and the next lecture to show kind of what these techniques are and how you apply them to different concrete data sets. So in terms of literature and useful references, for the image processing aspects, this image processing handbook is quite helpful. Again, you can get it online at ETH for free. And then for the models and ROC curves, there's very nice presentations from, one's from Julia Evans and the other one's from Stripe. So Stripe's an American company that does online payment processing. And so they don't do any image processing at all, but they use similar techniques to ensure that their models work well. So they do apply thresholds to determine if a transaction's fraudulent. And so they obviously have much more at stake than you do if they classify something incorrectly. And so it's a very interesting set of articles you can look at to get a better appreciation for these ideas. So kind of why do we do imaging experiments in the first place? So this kind of goes back to the first lecture a bit. There's this exploratory idea where we have samples we just want to understand better. We want to try to quantify, or we want to see what's going on inside the samples. We don't know what it looks like inside of a fossil. We want to see if a head is present or not. So this is where you have very little prior knowledge or expectations, and a lot of the work you're doing is very exploratory. The second sort of reason we do scientific experiments is really as part of the scientific method to test a hypothesis. And so we have some prior knowledge, we have some expectations, and we want to actually test to see if these prior knowledge or this prior knowledge is true or not. And so this is where we then want to have tools that help us quantitatively describe what's going on inside. And so some example questions, would be, does temperature affect bubble size? And so there you think, as the temperature goes up, the bubbles get larger because we've seen in some experiments that kind of appears. And so now we want to actually quantify how much larger it gets. We want to have control groups. We want to see how well that works. The same in biology, where if you're trying to figure out if a gene is important for cell shape and mechanosensation in a bone, then you can knock that gene out and measure and quantify these shapes and see how big the difference is, see how big the variation is, and determine if that's significant or not. Higher canal volume make bones weaker. Granule shape affect battery life expectancy. And so, of course, all of these questions aren't necessarily simple, and doing one experiment and measuring one value will certainly not be enough to confirm these hypotheses, but these are the kinds of questions that typically get asked. And this is why we need sort of more advanced tools than just image enhancement for answering these questions. And so what are we looking at in these images? So here's just a very kind of simple image of a bacteria. You see all these different components. In the textbook, everything's nicely colored. And so it's very easy to tell apart what's going on. And if you, use an imaging modality to take the same image, you now end up commonly with a black and white or grayscale image that you're looking at, where the values represent some sort of absorption that you're seeing in the image, so some sort of physical property. And so, to test a hypothesis, we want to perform an experiment on bones to see how big the cells are inside it, for example. We have this whole pre-processing step. So this analysis is covered in other courses, how you actually collect the data, how you do this reconstruction in the case of tomography data. We then do the image enhancement, which we talked about in this course. And so you have this really large data set with lots of different values inside, lots of noise, lots of sample. You do all the image processing techniques that were discussed by Anders last week. So all of these complicated filters, non-local means, Gaussian, everything else. This takes a very long time. And you end up with exactly as much data as you had before. And so if it didn't help you reduce the amount of data, why was this useful?
We have less noisy data.
Yes. So we have much less noisy data. The data is much easier to look at, and it hopefully makes the next steps that we perform much easier. And so we still have right now way too much data that we somehow need to reduce. And so if we look back at kind of what we wanted in the first place, this is a single number. A volume fraction, a cell count, the average cell stretch, cell volume variability. This is the kind of information we're trying to extract. And in almost every experiment you'll do, that's really what you're trying to get. You're not trying to get a tensor. You're not trying to get complicated other images. You're really trying to get a single number, because if you want to perform a statistical analysis, you'll need to take all of these single numbers and do something with them. So why do we perform segmentation? So in a model-based analysis, every step we perform, simple or complicated, is related to kind of an underlying model of the system that we're dealing with. And so if we go back to that bone sample, we have air, we have bone, we have tissue. We know that bone interacts much more strongly with X-rays than air or tissue does, and so we'd expect it to appear brighter on the image. That's kind of the model we have, and we want to apply that to the image. This is where aspects like sort of Occam's razor is very important, where the simplest solution is usually the right one. And so if you're dealing with something like bone, trying to incorporate every possible piece of information you know about bone when you're segmenting it will likely make it a lot harder to do your task, and you want the simplest approach you can take in order to segment it. And so what that means sort of concretely is that, rather than saying we expect bone to be exactly 355 Hounsfield units in density, and we expect the bone density to be almost constant, so we'll look for sort of large constant areas at 355. We'll probably find nothing. Versus if we say sort of generally bone should be between 300 and 600, and we'll include everything that falls in that area, that works much better. We will, of course, talk about some of these techniques later, but the idea is to start off as simply as possible and only add complexity later. So also with bones, if you're like, bone is really difficult to segment, I should train a neural network to do that. That could work well, but you're now training a much more complicated model that will fail in much more complicated, bizarre ways. And so unless you need a neural network to do that task, it probably doesn't make sense to segment bone with a neural network. We have some other slides where we've sort of done experiments with this, where you use a very common neural network architecture to segment something like bone or lungs. And basically it takes 41 million parameters that you need to fit. It takes about an hour to run on each sample. and it's worse than performing a few simple filters and thresholds. And when it doesn't work, so if you rotate your sample a little bit, it loses some of its perspective, and then you end up with sort of a completely wrong segmentation. And so that's a very clear example where neural networks are very good at solving very complicated problems, but they can be very bad at solving very simple problems. So we'll talk a bit more about these techniques, but we'll start off with the simplest ones because it's important to understand them and try to improve upon the simple techniques rather than starting immediately with the most complicated ones. So if we go back to sort of filtering and image enhancement, we have sort of these slides, which we covered a little bit before. These are sort of oversimplified equations where you basically have sort of what you measure is equal to your sample plus some amount of noise. And so what the perfect filter would be is you apply your filter to your sample, you get your sample out, you apply it to your noise, you get 0 out, and you apply it to your measured, and you basically get sort of just the real or sample signal out. And the problem is what most filters end up doing is they affect your sample signal, and they don't completely get rid of the noise. And so even good filters are going to reduce your sample signal a bit and not necessarily remove all the noise. And what really bad filters do is they reduce your sample and they don't really reduce your noise very much. So since we covered some of these things last week, I'll see if this works. So we added a new image enhancement tool. See if this still runs. It's again on the course website, and it makes some of these things that Anders talked about last week a little bit more clear. This is also all Python notebooks. You can click edit to see the source code if you're really interested. But you have a picture here. You have a type of noise, you have a noise level, a filter name, filter shape, filter size. So we won't really talk about the filter shape and size right now. But here you have an original image. You add some noise to it, like salt and pepper. You perform a filter, and then you look at what the difference is. And so what did your filter actually change in the image? And so if we do a Gaussian filter here, we can now see that we got rid of some of the noise, so this is not nearly as speckled as that, but we've also gotten rid of some of the signal. And so when you look at this difference image, you see structures appear, which means you've gotten rid of some of the signal. So that was not a perfect fit for this kind of problem. And so it damaged both the signal and the noise. If you now go up to median filter, you have almost no noise left, and you have very little signal present in what you're doing. You also have this sort of mean squared error term, which is what we use to kind of quantify how different from the original image does it look. And that we have, 0.09, which is obviously much better than 3.3. And so here you have the ability to play around with the different filters and tools and get a feeling for which filters work well with which kinds of noise. And so we have a number of different types of noise. This is another one where it's just sort of horizontal stripes. You see this often if you have a detector with sort of one of the sensors broken, that you'll end up with a bunch of stripes in the image. And that, again, this filter doesn't do a particularly good job at focusing on just the signal and getting rid of these stripes. And so that's where you require slightly more complicated approaches to deal with the fact that you have this sort of structured noise rather than just random noise. But yeah, feel free to look at this, and we'll probably come back to this a little bit later when we talk about neighborhoods. So in terms of quantitative metrics, what did people used to do? So if you go way back to kind of the original image that we showed before and what people were doing, I think even as late as the 70s, when you would take images like this, you would identify objects by eye. So you'd look at the image and you'd sort of say, This is a bacteria. It's got a cilia on its surface. It has a long curly flagellum, and a lot of biological papers you read today still talk in very much this way about the structures and the things that they see. If you want to start quantifying things, this is what happened sort of in the 70s, where you have images like this that you print out on equal weight paper, and then you cut it out well, and then you weigh it, and that will give you an estimate for what the area of the object is. And actually, if you go to the hospital today and you see a pathologist, and so sort of they take a biopsy and they look at slices from it, they will identify most of their job is entirely qualitative. So they might manually count some things on the slides, but the slides are never digitalized, and it's just sort of a picture on a microscope, and that's what they're using to diagnose you. And that works fairly well, but it's obviously time-consuming. There's obviously many chances for mistakes to be made, and it's not very easy to look at every aspect of the slide because you're a human and you can only look at one part. And so now we kind of come back to these topics from the first lecture, where we sort of have this different worldview and how they fit in. So we have sort of the experimentalist approach, which is what we've talked about for the most part up until now, and this is really sort of the model-based, where you have problem-driven, top-down, and what is the reality of your system, of your detector, what are the physics involved, and how can you come up with a model for segmenting or processing your data. We then sort of have the machine learning approach, where this fits for sort of computer vision and deep learning, and this is much more results-driven. And so we'll start off with sort of this model-based, and we'll show why the model-based isn't really sufficient for all problems that we deal with, and where the computer vision problems start to come in. So if we take the model-based analysis, we have sort of this slide again from the beginning. There's some kind of impulse. You're looking at a characteristic inside your sample. You want to sort of get a response out, and you're detecting this in some way. And so there's lots of different modalities which use fundamentally this principle. There's a lot of similarities in the underlying equations, and that basically you end up with your measured signal is some sort of function of your stimulus and your sample. And so whatever you're looking at might have different functions for that, but that's the basis. And so if we look at direct imaging with sort of a simple example, where you have a beam profile, so you might be using sort of a constant illumination if it's a microscope, a laser if it's a confocal system, or any number of other similar steps between, where you have your system is simply multiplicative, your stimulus is sort of the profile of the beam, and what you're looking at the sample is sort of this alpha term. And that the alpha term is basically your measured divided by your beam profile. And what we see for this is if we take this sample like before, and we have a beam profile that's not constant, we end up with an image at our detector which is not constant. And so this is one of the common things you'll see when you're doing image processing tasks, is that you end up with an image like this where the intensities don't really represent the sample profile anymore. And so when you're looking at the intensities, there's a lot more information about the beam than you want, because ideally you're not interested in the beam profile at all. All you're interested in is the sample. And so how can you start to separate these things, and how can you model it well so that you can account for the fact that that beam profile isn't constant? In X-ray imaging, one of the things that you do is you take the sample out and you take measurements with no sample at all so that you get a very accurate representation of the beam, and then you put the sample in, and then you simply divide those two. Can anyone think of why that would be problematic, or when that wouldn't work well?
The beam profile had any zeros?
Yeah, so if the beam profile has zeros, that information is lost, so you'll never get that information out again. That's good. Any other times that might be problematic? No ideas? So if the beam profile is not stable, so if the beam profile moves around, which again is something you get in X-ray imaging fairly frequently, that the beam doesn't stay constant, it evolves with time. I think some of the experiments we did had, I think, 200 hertz dynamics that were in the beam profile. And so it makes it very, very difficult to get an exactly constant image of the beam. And so even when you get it correctly, and even if there aren't any zeros, and often there are zeros, then you'll still have the problem that sort of dividing this won't give you exactly your original sample back. But there's some of the things you can start to do to correct for this fact. Because if you don't correct for it at all, you'll end up potentially analyzing more about your beam profile than about your sample itself. So here's an example where you sort of have a cut through of your sample where you can see the sample itself is this green line, the sample itself is this orange line, the sample profile. The green line is what your detector actually sees, and the blue line is your beam profile. And so you kind of see that the problem is you have these two things, or these three things all mixed together. And so it makes it very difficult to see, is this actually a flat constant region, or is there a small slope there? And particularly for questions like that, which could be important if you're trying to determine, where the nucleus is concentrated, or if the nucleus, if a cell's dividing, maybe the nucleus is sort of spread thinly in parts, and so there might be things like that going on. And if your beam profile is not corrected, it will be very difficult for you to say if that's an artifact of your imaging or if that's in fact part of the cell that you're looking at. Frequently there's fall off as you get away. With laser systems, this is something you always have to be aware of, that this is not a simple problem to solve. Here's the sample again. So for absorption imaging, X-ray, ultrasound, optical, we have this Beer-Lambert law. So has anyone heard of this before? Okay, good. So anyone who took Marco Stampanoni's class last semester, or any semester, he hammers this in quite well. This is sort of this relation, so that once we've taken this original profile, we talked about sample here. And ultimately, sample isn't really what we're interested in. We're not interested in how much the sample absorbs. We're interested in what the sample's actually made of. And so what this allows us to do is sort of break down. So here's the detector, here's the source, the stimulus. We have the sample, and the sample's this exponential decay with this alpha coefficient and this d, which represents a thickness. And really what this allows us to do is kind of go away from just a purely, this is how much a part of the sample absorbed, to we can make a full model for what's going on because we know d. If we have an idea of what the sample's made of, sort of the atomic number, the cross-section, so some coefficients that are quite easy to remember, we can then pretty accurately identify what material is in the sample based on this absorption image that we get. So for many samples, there are, if we're trying to segment this, if there's two or more distinct components, then we would expect alpha to have two sort of distinct values that we could associate with in order to decide how we're going to threshold and separate our image. And that's basically what this comes down to. Is this gives us the link from the purely imaging side of things to the physical or chemical side, where we can start to actually quantify what's going on and come up with some basis for our assumptions. And so this is exactly what we do with sort of the bone images, where we know what this alpha coefficient is for bone, we know what it is for air, and we can then determine if a part of the sample is bone or air because of our understanding of these coefficients. So here is kind of an example. Don't worry about the code at all, but basically you have sort of an alpha coefficient. You might have three different materials which have sort of the spread of alpha, and you end up with the Beer-Lambert law giving you this kind of exponential decay in what their actual values are. And that you see the alpha coefficients, which are shown here, are distributed more or less kind of uniformly, and the resulting intensity values, or eye detector at a constant intensity, sort of get much closer together as you get further down here. And so the result being ultimately that the relationship between alpha and what you actually see on the detector is not linear, which means that some things which are very close here will be further apart when you actually look at them on your detector, and some of the things that are very close together or well separated here become even more close there. And so just because your alpha values are different doesn't necessarily mean it will be easy to segment, because you have this sort of nonlinear transformation you have to take into account. So an example for this is mammography, where you basically have a detector, a source. Very commonly, you sort of have a breast. You have a X-ray detector on one side that shines through it, and then you make this 2D image of mammography. So I assume everyone's seen a mammography image before. I hope this isn't not safe for work, but I think it should be reasonable. So images like this, where you basically are able to look through a breast and find sort of suspicious things inside it. That's the whole goal of mammography. You use X-rays to do this, because X-rays give you the ability to kind of see through skin quite easily and differentiate some of the different tissues you have inside. And in this example, it's quite clear that you have sort of a lesion structure here that is very different from this fibrous material or fat that's present. And so this is something that's then quite suspicious, that's investigated in more detail. So the principles behind it are the same as what we've talked about so far. You have a detector, you have a source, you have this exponential decay. You have sort of a continuous sample. So in this case, it kind of becomes an integral, where you have different alpha coefficients at every point inside the breast. And then you have sort of a differentially small thickness along the projection axis that you integrate over. And so it's specifically quite difficult to separate this alpha and this D. And so if we take sort of a very simple basic breast volume with a half sphere and a constant absorption factor, so 1 times 10 to the minus 2, we can turn this into a sum in discrete space. You don't have to worry about this code again. This is just how we make it. And you now end up with this simple model of a breast. And so this is the 3D phantom we have for it. Obviously, this is a drastic oversimplification. Breasts don't, in fact, look very much like this, but it's a starting point, and it helps us illustrate why this is important. We can now simulate our detector, so we can apply sort of this Beer-Lambert's law with sort of the exponential decay as we sum over the breast volume, and we end up with this very simple mammography image. And so this works quite well. We see sort of what the shape of the breast is, and because it's homogeneous, everything sort of looks okay. If we know that alpha is constant, we can then reconstruct D from the image. And so here, we're able to take the fact that it's constant and apply sort of the inverse of the Beer-Lambert's law, where we do this sort of separation. We now have the thickness of the breast at every point. We can then represent that as sort of a 3D plot, where we do get an accurate reconstruction of the shape based on the information that we have, or at least half of the shape. So we run into problems, of course, when alpha is no longer constant. So if you have a dark lump in the center of the breast, which is, again, exactly what these physicians are looking for, and you have some unusual patterns inside of this, and so here we have a fairly construed situation where we have a very oddly patterned lesion inside of the lump. And so if you sliced the breast apart, slice by slice, it would look something like this. And Problem is, because we are just doing a projection through this whole image, everything gets kind of stacked on top of each other, and the alpha and the D get mixed together. And so we're no longer able to reconstruct that information. And so what we end up with is a constant region here, because when you sum all of those things up, some parts are brighter, some parts are darker, some parts are thicker, and you end up with just sort of a constant flat region, which, looking inside it, is not at all what you would see. You would see much more detailed, interesting pathology. And so if you try to reconstruct the D values from it, you would sort of end up with a oddly shaped breast here, when in fact the breast shape is perfectly normal. All we have inside is a very odd lesion. And so this is sort of one of the issues, obviously a very invented scenario, but it's something that radiologists deal with a lot, that these projection images don't provide enough information to be very certain. And particularly when you're dealing with breasts that do have very complicated structures inside, it can be a lot harder to tell if something is part of the breast or if something's a cancerous lump or a lesion that you need to be worried about. And so while these techniques work very well, you end up with a lot of problems of trying to separate these things in order to sort of segment what is the breast, what's the lump, and everything else. And so now, that was a little slight detour from segmentation. And so what segmentation gets us is that we're able to take sort of all of these images, everything that we've measured, and now start to group them into different groups. And so for the images we were just talking about, the absorption ones, you'll want to take sort of this alpha value and put it into a, breast, non-breast, or tumor, non-tumor group. For red, green, blue images, you might want to take them and put them into kind of a, person's face, not a person's face. Spectrum or hyperspectral imaging. You might be trying to, look at satellite images of the Earth and decide if something is a cloud or a glacier, which is one of the problems they struggle with a lot in these images, and that's where spectral information is very helpful. Or if you're looking at sort of a mechanical stress field, you might be looking at a vector or a tensor as an input. And basically what this means for these samples that we talked about before, after the filtering, you ended up with just as much data as you had, but much less noise. That if you do this thresholding, you now end up with sort of 1 bit at each position. So you make a decision for each pixel or each voxel in this data set if it's in the foreground or in the background. And that's the main classification we'll focus on now. Obviously, if you're making multiple groups, then this isn't 1 bit anymore, but it's a very small number of bits. It's a very small, you don't end up with thousands of different classes. You end up with maybe 5 or 10 at a push. And so if we look at how do we apply a threshold to an image, we take a sample image here. This is just a cross. You see the image there. There's a range of values that go inside it. It's not particularly exciting. For us, what's important to see is that there is a cross and there is some noise in the background. And so how do we separate the cross structure from the background? So we can represent the intensity in sort of a probability density function. And this is where we make a histogram of all the intensity values that we see. And so here, there we have each pixel in this image that has a value between -0.2 and 0.8. And here we see between -2.8, and we have the number of pixels that fall into any one of these values shown there. And so this is a continuous range. In order to make a histogram, we need to decide on sort of a bunch of small buckets. So maybe -0.2 to -0.3, -0.3 to -0.4, something along those lines. And then we count the number of sort of pixels in each bucket, which gives us a probability distribution function for what's inside our image. And so what this enables us to see is kind of where the pixels group together and where we might have a separation between this cross and the pixels we see before. And so from that, we can then apply a threshold. The threshold's, again, very, very simple model where we say 0.4. And so if we go back, 0.4 was about there. And so it looks like these points could potentially be part of the background or part of the foreground, but fairly reasonably between. So we can then apply that. And we then see we get all the pixels in the cross, but we end up with 2 pixels included that aren't supposed to be in the cross. And so we could change that threshold and see if we do a better job. I think all of these codes should be interactive. They should be interactive for you as well. Since the images are randomly generated, they'll probably look slightly different every time. But if we run this again, yeah, now you see it's a different random image, so it gets one wrongs in different places, and we can maybe try making this 0.45 so that it gets fewer images. And there, it does a much better job, so we only have 2 incorrectly classified pixels. If we then do 0.5, we have looks like no incorrectly classified pixels. So 0.4 was too low, but it was a starting point for what we were trying to do.
Why does the image have more than 2 colors? I thought we just say 0 or 1 if the sample is in the threshold or not.
Yeah, so we still show the intensity in the background, just for sort of reference. The 0 or the 1 is the green.
Oh, okay, so you just color it a little bit.
Exactly. So what we do is we say, if you look at the code that we have here, basically it just takes the images, the values that are above 0.4, and it plots a square at each value that's above 0.4. And so if we were, we can do that quite easily. Instead of doing this, or actually we can just do cross image. We can just do that, and we end up with the image without any of the color values. We just keep the color values because sometimes it's unclear why one pixel got put in one category. And so when you can see the underlying color values, it's a little clearer, but yeah, it can be confusing. Obviously, when you take the threshold, all of those different colors are gone. It's just white or black. We can also see the effect of kind of taking a number of different thresholds where, if you took a value of 0.1, you would have way too many pixels. If you took one of 0.9, you would have way too few, and that there's this sort of trade-off between getting too many false positives and too many ones that you've missed. And so that's one of the things that we'll talk about. And in this image, we have a perfect threshold. So it looks like, 0.5 is this perfect balance. But if you can imagine that this noise changes every time, sometimes it gets a little bit brighter, sometimes it gets a little bit darker, you wouldn't necessarily be comfortable hand coding 0.5 in and hoping that that value magically worked forever. And so a lot of times picking this value isn't just about knowing, okay, the cross absorbs more than the background. It's about sort of making a trade-off between a number of different factors that are important to pay attention to. So segmenting cells is then sort of a more clear example of this. This is called white field microscopy, where you have some sort of illumination. Again, you have a profile that probably needs to be corrected for. We don't do that as part of this experiment. And you end up with an image like this. And so you have dark spots where cells are and bright spots where the light just transmitted all the way through. You end up with this histogram. And you can see by looking at this, well, what do you see by looking at that? In terms of segmentation. Does it seem like it's very easy or very hard? And why? Yeah.
It's rather hard because there's no bimodal distribution by the tail.
Exactly. Exactly. So when you have kind of peaks, it's very easy to kind of be, okay, that one peak is probably the light, and that other peak is probably the background. But when you have just one giant peak here, it's very difficult to say exactly where that cutoff should be. And so what should you do to divide between these two groups? And there's not a very clear decision to make because it is a tail. And so where exactly you put this tail in will affect what you see. And so we see the same thing in these images, where if you take a number of different thresholds, we show these values as red overlays. I guess that's not super clear on the projector. It should be clearer in the slides. Alternatively, we can just run this again. So this is with the overlay of the image. If we just do if we just show it this way, you can now see clearly what was counted as foreground and what was counted as background in each one of these slides. And even as a biologist, it's probably not particularly easy to say which one of those cutoffs makes the most sense. Because even in sort of the lowest one where you're missing a lot of cells, you have quite a few spots which are just dust that you want to ignore anyways. And so there isn't really a perfect value for this, but presumably somewhere between 100 and 200, the right value is. And so you can use some of your information and model-based approach to start with the problem, but it's probably not going to solve everything for you. So for other image types, if you have image types that are made up of a number of, that have more than just one value, so this would be red, green, blue images, this would be vector fields that you measure, you have the same kind of issues to deal with segmentation. And so here we have sort of a vector field, you can zoom in, where you have sort of an X, a Y, an intensity on the detector, an X vec, and a Y vec. And so if we look at sort of what's called a pair plot of all of these, we can kind of see the relationship between them. It's easier to view that as sort of a field, where we have this plot where we can see at each pixel what the orientation of that vector is. So this isn't a particularly complicated image, but this is something you can often get from sort of strain or mechanical experiments, where you end up with that at every point. And there you still want a threshold because maybe you want to classify the volume of area that's being compressed versus the volume that's being stretched. And so we do the same sort of thing. You apply this kind of simple rule where you decide what your criteria are. And then you apply it to kind of each row, and then you end up with sort of this white section. So for the ones that are positive in X and Y, we want to include them in a region. And that the fact that the image is a very different kind of image and that you have multiple values or very high depth doesn't change how you deal with thresholding at all.
So you are taking the absolute value of the vectors?
Nope. What we take there, we take the X vec greater than or equal to 0.25 and the Y vec. So you could also take the absolute value. So you could apply a different rule to it. That's quite easy. So if we go here and say, so if we wanted to apply the absolute value, we have to, I mean, there are functions that do this, but we can define it ourselves. And so now we can say that sort of the square root of the square of X vec plus square root of square Y vec is, so the slides, if you hadn't noticed, have to be run for each vertical stack in order. So if you want to run any of the code there, you should run So obviously none of them meet that criteria, maybe 0.1, or all of them do. NP squared XP. Oh. So I think they're all unit length. And so if you take the absolute value, you get the same thing for all of them. So yeah, potentially that's not necessarily the most interesting thing to do. But of course, these rules that you come up with, that you can apply any transformation that is relevant for the task that you're trying to do. With the main point being is that a lot of times people run into more complicated data sets like this and just assume intrinsically that they must be much harder to deal with or require completely different techniques. Well, in fact, the same exact techniques you use to apply to a color image or a microscopy image, you can apply easily to a vector field or tensor field image, because it's the same rules you should apply, the data sets are very similar, and you can just use that starting point for dealing with it. This can also then be shown as a joint probability distribution where, like we had the histograms before, when it was just one-dimensional values or depth that we were dealing with, we now end up with a 2D histogram. And so instead of saying this is the line we cut off at, we have to pick a line in this direction and then pick a line in this direction, or a criteria in both directions for deciding what gets included into our image and what doesn't. And so it's a slightly different kind of problem. Here, the color represents the number of pixels that fall into each category. And so as we see here, they're all unit length. And so you kind of have around the edge a large number. In the middle, you don't have very many because they're all just pointed. It's all just unit length vectors that have different orientations. And so here, yeah, you can visualize that a little bit if you're familiar with 3D or multiple dimensions. Once you get past 2, it gets a lot harder to visualize, but you have the same approaches that you use to deal with them. So yeah, so there's an example where you can do a dot product with sort of the x direction to see if it's perfectly perpendicular to the x-axis. And you can also do arc cosine, any other number of transformations. It doesn't necessarily need to be a line. And particularly when you're dealing with vectors, it's likely that it won't always be lines. So are there any questions on that? Otherwise, I think we can take a short break and switch to the next. Okay, so I think we'll have a break, and then we'll go on to the next slide set.